everyone, and welcome to our first talk in macronutrients for human nutrition. And this week we're going to talk about metabolic regulation and cell signaling. Now, this course is a big course. There's lots and lots of content in it. And next week we're going to start talking about specific macronutrients. We're going to start with carbohydrates and then move into lipids and proteins. And of course, when we talk about macronutrients, it's a big process from consumption, digestion, absorption, transport, but a big part of what we're going to talk about is metabolism. And this is kind of the most complicated part and kind of the most abstract part of this course. So I really wanted to take this make an introductory lecture where we could introduce some of these concepts about metabolism that we're going to see throughout the semester. And I'm hoping that by giving this introduction, then when we get to specifics, the understanding will go a little smoother. So we're going to talk, when we talk about these metabolic concepts, what we're mostly going to talk about is chemical messengers and how these chemical messengers affect biological processes. And our last objective is to discuss the different types of membrane proteins and understand how these are involved in metabolic regulation. Okay, so when we talk about macronutrients, again, in a nutshell, what we're talking about is how does the food we eat, how is that incorporated into into the materials that make up our body, and how is it used for energy? Now, we know throughout our entire education in dietetics that our body really strives to maintain homeostasis, which means that our body has to keep levels of certain nutrients and other molecules in a really tight zone in order to ensure optimal biological processes. So, for one example that we'll refer to throughout this talk is insulin and glucose. So we know that our bodies really work really hard to maintain a really tight level of glucose in our bloodstream. So it wants to maintain this homeostasis. Now, that gets a little bit complicated because most of us do not have a glucose drip going 24 hours a day. So we're not getting a constant supply of glucose. So we're talking about glucose and carbohydrate in this example, but you can think about this in terms of any macronutrient, really. Our food supply is not continuous, and yet our body needs a continuous supply of these nutrients in order to make cells and have energy. So therefore, our body has to have mechanisms of maintaining our nutrient supply and maintaining homeostasis. And because we're not eating continually, we have to have mechanisms for storing fuel so that we can use it later when we're not eating. This slide kind of gives two figures that kind of show what we're going to learn in macronutrients in a nutshell. So here's the whole process that we're going to talk about for each macronutrient, okay? So we have an intake of carbs and fats and proteins. These are digested, absorbed, and transported, and the compounds are incorporated into our cells. These compounds are then used to make components of our body through biosynthetic pathways, or they can be used as energy or as fuel or incorporated into fuel stores so that we can use energy later. So you can see that this is a very complex, long pathway that involves many biological processes. So how are these regulated, right? Especially when we think about what we just talked about, that our food supply isn't continuous. So not only does our body have to regulate all of these processes that are going on, it has to adapt to its environment. Have I eaten recently? Have I been starving for a long time? Am I on a diet? Have I exercised? Am I inactive? Your body constantly has to adjust to these different conditions. So in a fed state, our body is going to try to really store up our fuel, and so that we can use it for when we are not fed. So it's going to store up our fat and our glycogen and our protein. But then as we move towards periods where we don't have any nutrient intake coming in, then our body is going to break down these stores or oxidize these stores so that we can use them for energy. So again, we have these really complex set of biological processes that always need to adapt to changing conditions. So how does our body do this? How does our body regulate our metabolism to keep us in this tight homeostasis? Well, really, these processes are regulated by a series of signals. Okay, so here's a figure from your textbook from chapter 22, and it's just a really simplified diagram of the control of energy balance. Okay, so let me walk through this and then I'll give an example. So here we have our system energy balance. So let's say we're in negative energy balance. I haven't eaten since breakfast, it's dinner time, I skipped lunch, I was really busy. Okay, so as we might recall, when we're in negative energy balance, what's one of the hormones that's going to predominate in our body? Glucagon, right? So that's our hormone. So our system is in negative energy balance. So our pancreas secretes hormones. This hormone is sensed by our central nervous system, and then our nervous system says, "Oh, whoa, your body's saying it's hungry and you haven't eaten in a while, so you need to change your food intake or you need to change your energy expenditure so that we can get back in system balance." So in this example, our CNS would say, "Hey, start eating something or stop exercising. We got to get our system back in balance." And so we have this kind of regulation process in which our GI tract and our pancreas and our adipose tissue send these signals that are then perceived by our central nervous system, which then tells our bodies what to do so that we can then maintain some system balance. Okay, so when we're talking about how we perceive these signals from the outside of our cell and have them have an impact on our cell, so how do we get these external chemical messengers to have an actual effect on how our cell acts? Now, if we think about it, we have our cell with its internal environment, and then we have its external environment, right? And what separates the external environment from the internal environment of the cell? How do these signals get into the cell? Well, it has to cross the membrane. So I think we're all very familiar with this little graphic here. So this is our cell membrane, and we know that it is a phospholipid bilayer, right? Which is a beautiful design in many ways that we'll talk about this semester. So our phospholipid bilayer has a phosphate group on the outside, right? And this is hydrophilic. So it's next to the water and next to the interior of the cell. And then it has this lipid tails, which face each other on the inside because they're hydrophobic. And because we have this phospholipid bilayer with this hydrophilic layer and this hydrophobic layer, it really takes a lot for a signal to get from the outside of the cell to the inside of the cell. So how does it, which is great because we really want to regulate these signals. We don't want to have our bodies go overboard in reacting to a chemical messenger, and we don't want them to not be able to react to this chemical messenger. So we have these really tight regulations of what chemical messengers can impact the interior of a cell. And here we have another really nice picture here of the lipid bilayer, but it's a little bit more detail here. So what are some of the things that we see? So let's look at this for a minute. What are some of the things we see within this phospholipid bilayer, right? So here we still have our bilayer with our hydrophilic phosphate and our hydrophobic lipid tails here, right? And they're facing each other. But then we have proteins that are embedded into this lipid bilayer and carbohydrates. And these have really important functions, okay? And we're going to talk about the different functions of the proteins. But first, let's just look at these. So here we have protein, it's called an integral protein that kind of spans the length of the phospholipid bilayer. We have these peripheral proteins here that don't span the whole length of the bilayer, but are just kind of on the edge of one side. We have, again, an intracellular or integral protein here, but this has a channel. This is a transport protein. We'll talk more about that. And then we have what we call here like a glycoprotein. And what this just means is It's a little carbohydrate chain attached to some of these integral or peripheral proteins. Okay, so membrane proteins can have different functions, and they have three main functions. Okay, so first we have our transport proteins, our receptor proteins, and our enzymatic proteins, all of which can be embedded in the lipid bilayer, and all of which play a really important role in the communication between the outside of the cell and the inside of the cell to maintain homeostasis in this cell. Okay, so we're going to kind of do a little overview of each of these, and then we'll look at each one specifically. So first we have these transporter proteins, and it's exactly like it says. It regulates the flow of nutrients in and out of the cell. So nutrients are transported in and out of the cell. Then we have our receptor protein. Our receptor proteins modify the cell's response to the environment. So here we have a chemical messenger of some sort, and we will talk about chemical messengers more. But let's say it's a hormone, let's say it's insulin or glucagon, and it lands on this receptor protein. And what happens is it changes the shape of this receptor protein so that then this protein on the inside of the cell has an effect on the cell's biological processes. So the receptor protein's just hanging out, the chemical messenger lands on it, changes how the receptor protein looks, and then that has a signal to the inside of the cell. And then finally we have our enzymatic proteins, which are probably very familiar to us, which catalyze biochemical reactions. So they're sitting on the edge of the cell, and we have a substance of some kind that comes up to the enzyme, and this catalyzes a reaction in order to transform the original substance into something new. We already talked about integral versus peripheral proteins. And one thing that we should note is that these proteins are embedded in this phospholipid bilayer, and of course it looks very stable here, but really these proteins are moving across the lipid bilayer at any time. And really the properties of the phospholipid bilayer can impact the fluidity of the membrane, and that can impact how these proteins are moving and how they're functioning as well. And we'll talk more about that, especially in our lipid section. So I talked about chemical messengers. Let's talk more about these chemical messengers. So it sounds like a really broad category, and it is. A chemical messenger has 5 specific properties, okay? And here's a really simplified diagram of one. So a chemical messenger is secreted from one cell in response to a stimulus. So let's go through these, and then I'll give a specific example. It also diffuses or is transported through the blood or other fluid to a target cell. The receptor on the target cell specifically binds to that messenger. The binding of that messenger then elicits a response, and then the signal ceases or is terminated. So these are kind of the 5 properties of how a chemical messenger works. So let's give an example. So a chemical messenger can be a protein, like we said, or a hormone, like we said, such as insulin. So let's see if this meets the criteria for a chemical messenger, insulin. So it's secreted from one cell in response to a stimulus. We know that's true, right? We know that when we have a lot of glucose in our blood, it stimulates our beta cells to produce insulin if we are healthy, non-diabetic. And so it releases insulin in response to this hyperglycemia, the stimulus. Okay, great. Then so it meets that criteria. It diffuses or is transported through the blood to a target cell. Yes, we know that. We know that insulin travels in the blood. We can measure hyperinsulinemia or hypoinsulinemia, and it goes to our target cell. So let's think about like a muscle cell, for instance. So it meets that criteria. The receptor in the target cell specifically binds to the messenger, so we know we have insulin receptors on our muscles, check. Binding of the messenger to the receptor elicits a response. Yep, we know that when insulin binds to its receptor, it increases the amount of glucose that can come into the cell, right? And then the signal ceases or is terminated. Right, it's not going to perpetually bring the glucose into the cell. It stops when the signal stops. So when our blood glucose gets to a normal level, the signal stops, the insulin is not released, and we've met homeostasis. So this is all of the properties of a chemical messenger. So here's a nice little graphic. Again, that's just describing what we just saw. So we have secretion of a chemical messenger from a secretory cell. The chemical messenger then is diffused through the blood or another fluid. It goes through the blood or another fluid, and then it can diffuse through the cell depending on what the chemical messenger is. More often, it binds to a receptor, and this binding to this receptor then has an impact on the cell's biological processes and elicits a response. So why are we going through all of this? What we're trying to do is establish kind of a conceptual understanding of why biological processes occur in metabolism. So we can talk about the Krebs cycle and TCA cycle all day, and don't worry, we will. But we need to know what is initiating these biological processes. And it's really these chemical messengers that respond to changing environment in our bodies. Okay, so for the chemical messengers, we have a few different types. So first we have, and they each represent a different system in our body. So first we have the neurotransmitters and neuropeptides. So this is part of our nervous system. An example of this is epinephrine. So this should be a pretty familiar one to you. We know epinephrine is a neurotransmitter from a stress response. We also can have hormones can serve as chemical messengers. And this is the area that we're going to talk about the most. So hormones are part of our endocrine system. And some examples that we will talk about a lot are insulin, but also epinephrine, in addition to being a neurotransmitter, is also a hormone. Our steroids are hormones. Our thyroid produces hormones. So first, I have a couple of videos here that are actually going to take a little bit of time, but the first one we'll talk about, that we're going to watch, just gives you kind of an introduction to hormones. So when we think about hormones, we may think of certain things, but really these hormones are what is dictating how all of our metabolism is regulated. So let's look at that.
Let's suppose that you're writing a really important email to a colleague.
I've invited you all here today because I wanted to talk to you about some ugly stereotypes that are going around. I've been hearing a lot of unfair, unseemly, and unscientific generalizations being made lately, and they mostly have to do with sex and your hormones. People have a nasty habit of equating hormones with a particular set of behaviors and conditions, most of which have to do with reproduction or sexual development or acts that include what my brother John has referred to as scootily pooping. For example, people will say that hormones are why Kevin has zits and is being all moody, or why Hannah, who's 3 months pregnant, just cried watching a commercial for car insurance, which, let's be honest, I do that too. Now, I'm not saying that hormones aren't at the root of sexual attraction or zits or occasional bouts of extreme emotion, because they are. That's just not all that they do. Not even close. When people talk about hormones in the context that I just mentioned, what they really mean is sex hormones. But sex hormones are just one kind of hormone that you have coursing through your body right now. In fact, there are at least 50 different types of these chemical messengers at work in your body at this very minute, but only a very few of them have anything at all to do with sex. The truth is, from birth to death, just about every cell and function in your body is under your hormones' constant influence. They're floating through your blood, regulating your metabolism, your sleep cycle, your response to stress, and the general and incredibly important overall homeostasis that keeps you not dead. Some hormones are just there to make other hormones trigger even more hormones in a kind of chemical relay race that biologists refer to rather elegantly as cascades. These hormones run through your body no matter what your mood is or whether you have zits. So the reality is, we are all hormonal all the time. Okay, to begin to understand our hormones and the endocrine system that produces, releases, and reabsorbs them, we have to step back and take a broad view. Not just by emphasizing that sex hormones are not the only hormones you have, but also by looking at how your hormones interact with your other organ systems. Because if anything, your body has two bosses, two complementary systems that are constantly shouting instructions over each other to all your bits and pieces. Both your endocrine system and your nervous system are constantly trafficking information around your corpus, gathering intel, making demands, controlling your every move. They just have totally different ways of doing it. Your nervous system uses lightning-fast electrochemical action potentials delivered by an expressway made of neurons to specific cells and organs. But your endocrine system prefers a slower, wider stream of data. It secretes hormones that travel through your blood, not through neurons, so they move more slowly, but they also produce widespread effects that last a whole lot longer than an action potential. Now, compared to your heart or brain or other arguably more glamorous organs, your endocrine system's organs and glands are kind of small and lumpy. They're also rogues. Instead of being all nestled together like in your other organ systems, these guys are scattered all over the place, from your brain to your throat to your kidneys to your genitals. A gland is just any structure that makes and secretes a hormone, and the master gland in your body is the pituitary, which produces many hormones that signal other glands, like the thyroid, parathyroid, adrenal, and pineal glands, to make their own hormones. The endocrine system also includes a few organs, like the gonads, the pancreas, and the placenta in pregnant women, all of which have some other non-hormonal functions and are made up of multiple tissue types. And technically the hypothalamus in your brain is in the endocrine club too, since in addition to all of its busy brain duties, it does produce and release hormones. So thanks to all these glands and organs, you've got these hormones diffusing through your blood doing all sorts of different things, but the thing to remember about them is that a hormone can only trigger a reaction in specific cells. They're so-called target cells that have the right receptors for it. So just like some keys can open many locks while others only work with one, so too can the hormone-target cell relationship either be widespread or localized. You're probably going to want an example of that, so your thyroid at the bottom of your throat produces the hormone thyroxine, which stimulates metabolism and binds to receptors in most of the cells in your body. But your pituitary, which is nestled all comfy under your brain, produces follicle-stimulating hormone, which helps regulate growth and trigger sexual maturity, and it only targets specific cells in the ovaries and testes. So, how do hormones bind to their target cells? Well, chemically, most hormones are either made of amino acids, including their more complex structures like peptides or proteins, or they're derived from lipids like cholesterol. And this is key because a hormone's chemical structure determines if it's water-soluble, like most amino acid-based ones are, or lipid-soluble, like steroids are. Solubility is important because your cell membranes are made of lipids. That means that water-soluble ones can't get across them, so target cells for those kinds of hormones have receptors on the outside of their membranes. Lipid-soluble hormones, on the other hand, can just basically glide right through that cell membrane, so their receptor sites are inside the target cells. Either way, when a target cell is activated, the hormone alters its activity by either increasing or decreasing some of its functions, usually with the goal of maintaining your body's homeostasis in one way or another. So, if hormones are keeping your body in balance, what's putting your body out of balance? I don't know. Could I interest you in some pie? If you have a couple of nice, generous helpings of strawberry rhubarb pie, and just to make things interesting, let's say they're a la mode, your blood glucose level is going to go through the roof. And the pancreas regulates your blood sugar by releasing two different hormones, insulin and glucagon. Once you have a belly full of that pie, beta cells in your pancreas release insulin, which helps lower your blood sugar by increasing the rate at which your cells store the sugar either as glycogen or as fat for later use. Now let's say you've done the opposite. You've eaten no pie. You're pieless. In fact, you've eaten nothing for hours. If your blood sugar drops too low, then alpha cells in the pancreas will instead send out glucagon, which helps raise your blood sugar levels, in part by decreasing the storage of sugar in your cells and triggering the release of glucose back into the blood. Lots of different endocrine-related illnesses, like diabetes or hyperthyroidism, tend to be the result of either hyper, too much, or hypo, too little secretion of certain hormones, which throw your homeostasis off balance. But there are lots of more common and less obvious ways your hormones can get out of balance, not because of some disorder, but because these signaling chemicals are just caught up in a chain reaction, which can take a while to subside. Some hormones just exist to control other hormones, which in turn control still more hormones. So as soon as one starts to trickle out, you can pretty quickly wind up with a cascade on your hands. You got a few different hormone cascades going on at any given moment, but one of the big ones, one that's really worth understanding, is the hypothalamic pituitary adrenal axis, or the HPA axis, because you don't want to have to say that every time. This is a complex series of interactions between three glands that ultimately regulates lots of your body's daily processes, like digestion, sexuality, immune response, and how you handle stress. And it's complex not just because of all the glands involved. It's also one of the more crucial instances of your endocrine system coordinating with your nervous system. Specifically, it's behind that fight-or-flight response that everybody keeps talking about. The HPA axis is essentially the endocrine system's companion to the sympathetic nervous system. The sympathetic system, in times of high stress, does things like speed up your heart rate and direct blood away from your digestive organs and to the muscles. But many of the other effects of the stress response are carried out by your endocrine system. And getting your nervous and endocrine systems to work together in times of crisis is where the hypothalamus comes in. It's the hub of where the two systems meet. It keeps tabs on what's going on all over your body, analyzing your blood for signs that something might be off. So let's revisit our fight-or-flight scene from a few lessons ago, the old burning house scenario. So you're sleeping. You're dreaming about petting pandas with Emma Watson or whatever when the smoke alarm goes off. Well, action potentials in your brain trigger neurons in your hypothalamus to release the peptide hormone CRH, or corticotropin-releasing hormone. The CRH makes the very short trip through the bloodstream to the anterior pituitary gland where, because it is water-soluble, it binds to receptors on the outside of its target cells. There, it triggers the release of adrenocorticotropic hormone, or ACTH. The ACTH travels again through the bloodstream to the adrenal cortices of the adrenal glands on top of your kidneys. When the ACTH binds to receptors on cells in an adrenal cortex, it triggers the release of a frenzy of different freak-out compounds known as glucocorticoid and mineralocorticoid hormones. Typically, these hormones help us deal with day-to-day stress by keeping our blood sugar and blood pressure balanced. But under major stress, like waking up in a burning building stress, these hormones, like cortisol, cause the classic fight-or-flight response, ramping up your blood pressure, dumping glucose into your bloodstream, shutting down non-emergency services like your immune system and sperm and egg development. And guess what? Now that all these stress hormones are pulsing through your blood, the hypothalamus back in the brain senses them. And because its job is to monitor and maintain balance whenever possible, it then stops secreting CRH, which eventually causes the other glands to stop secreting their panic hormones. Now, because this element of the stress response is hormonal rather than electrical, it comes on more slowly than the nervous system part, and it takes longer to subside too, as those stress hormones linger in the blood before being broken down by enzymes. So, we're a long way from teenage crushes and zits and crying over commercials at this point, aren't we? As a lifelong owner of hormones, I hope you'll join me in dispelling the stereotypes that surround these powerful and important chemicals and give them the respect they rightly deserve. Today, we looked at the endocrine system and how it uses glands to produce hormones. These hormones are either amino acid-based and water-soluble, or steroidal and lipid-soluble, and may target many types of cells or just turn on specific ones. We also touched on hormone cascades and how the HPA axis affects your stress response. Thank you to our headmaster of learning, Thomas Frank, and to all of our patrons.
Okay, so what he talked about is this kind of hormone cascade. So we have a signal from our hypothalamus that goes to our anterior pituitary, then to our trophic hormones and target glands. These target glands then secrete hormones and then lead to inhibition. So he talked about this downward cascade, but then their hypothalamus can then, and our pituitary glands can then sense that these hormones have increased, and then they stop producing it. So that's called the negative feedback inhibition. So I do now, the chemical messengers that are of the greatest importance of this course, I'm sure you got it already, are going to be insulin and glucagon. And so I want to take a specific look at those in this introductory talk. And this one is not as interesting as the last one, just to warn you.
Today I'll be discussing the normal physiology of two of the body's most critical hormones, insulin and glucagon. This video can be viewed in two ways, either as a standalone physiology lesson or as the lead-in to a forthcoming series on diabetes. By the end of this video, you'll be able to describe the macroscopic and microscopic structure of the pancreas. You'll be able to list the major metabolic actions of insulin and glucagon. You'll be able to describe the regulation of insulin and glucagon, including the role of other hormones, for example cortisol, epinephrine, GLP-1, and GIP, among others. And last, you'll be able to describe the structure and synthesis of insulin. A few things that will not be covered in this video include the structure of the insulin receptor, glucose transporters, signal transduction mechanisms, and the individual enzymatic steps where insulin and glucagon act. While these details may come up on a biochemistry exam in med school, their inclusion would add another 20 minutes, and these topics are almost never of clinical relevance. I'm going to start off by mentioning 3 very pervasive myths about insulin and glucagon. The first myth: the only major function of insulin and glucagon is to regulate blood sugar. Second, that insulin and glucagon are the only significant hormones involved in glucose homeostasis, specifically the very simple model in which insulin makes the blood sugar go down and glucagon makes the blood sugar go up. And last, that modern medicine has a complete understanding of the actions and regulation of these hormones. Despite the fact that I encounter students and doctors alike with these beliefs, none of them are true. Insulin and glucagon do much more than regulate blood sugar. There are many other hormones involved in the regulation of glucose. There are unequivocally 7 other hormones with a clinically relevant role in glucose homeostasis, and quite likely more whose role is waiting to be further elucidated and confirmed. Which brings up the last myth. While we understand much about insulin and glucagon, modern medicine is gaining new insight into their very complex physiology every year. So now, where do we begin with what we do understand? Well, a good place to begin is with the pancreas. The pancreas is a glandular organ in the upper abdominal cavity located posterior to the stomach. It is typically divided into 3 geographic regions: the head, which is nestled against the duodenum, the body, and the tail, which stretches laterally towards the spleen. A network of ducts coalesce and exit the pancreas via the main pancreatic duct, while the common bile duct brings bile down from the gallbladder and liver above. The pancreatic duct and common bile duct merge just prior to an opening into the duodenum called the ampulla of Vater. A significant minority of patients have an accessory pancreatic duct, which drains proximally to the ampulla and is of minimal clinical relevance. When viewed microscopically, the interesting nature of the pancreas becomes more apparent. Here are 2 microscopic views. A histology slide is on the left and an artistic rendering on the right. We can see that the organ appears to be composed of 2 discrete types of tissues, each of which corresponds to one of the pancreas's 2 major physiologic roles. The first tissue type is the acini, or acinus when singular. The acini are responsible for the pancreas's exocrine function by secreting digestive enzymes into the duodenum via the network of pancreatic ducts. The second tissue type are the islets of Langerhans. The islets are responsible for the pancreas's endocrine function. Instead of secreting enzymes into the gut, they secrete hormones into the blood, most notably insulin and glucagon. They're named after Paul Langerhans, a German pathologist and physiologist from the 19th century, who first described them when he was a 22-year-old student. The islets are composed of multiple cell types, each of which secretes a different 1 or 2 hormones. Alpha cells secrete glucagon. Beta cells, which are the most numerous, secrete both insulin and another hormone called amylin. Gamma cells, which are frequently referred to as PP cells, secrete pancreatic polypeptide, the function of which is unclear and which won't be discussed further in this video. And last, delta cells, which secrete somatostatin. A 5th, much rarer cell type has recently been discovered and is predictably called epsilon cells, which appear to secrete a hormone called ghrelin. As already implied, insulin and glucagon play a critical role in maintaining metabolic homeostasis, and they act largely in an antagonistic way such that the primary actions of one hormone are the complete opposite of the primary actions of the other. When the body is in a well-fed state, insulin predominates. The primary actions of insulin are glucose utilization, known as glycolysis, glucose storage as glycogen, known as glycogenesis, fat synthesis or lipogenesis, protein synthesis, and general promotion of glucose uptake in tissues, which also helps to promote the previous 4 actions.
We'll talk about
These all make sense. When the body is well fed and fuel and energy is abundant, of course your body should store excess energy. When the body is fasting, however, glucagon predominates. Its primary actions are glucose synthesis, called gluconeogenesis, glycogen breakdown or glycogenolysis, fat breakdown through a combination of the processes of lipolysis and beta oxidation, protein breakdown, and last, the formation of compounds called ketone bodies, which act as an alternate fuel source when glucose is in short supply. This balance between insulin and glucagon, and between the well-fed and fasting states, is occurring constantly, being regulated on a minute-to-minute basis, based upon the relative abundance or lack of nutrients in the blood, most notably, though not exclusively, glucose. The primary goal of this balance is to provide a constant level of available fuel in the bloodstream, irrespective of how recently a person has eaten. And in the absence of diabetes, extreme physiologic stress, or rare hormone-secreting tumors, the body does a fantastic job maintaining euglycemia, that is, a normal serum glucose level. I'm going to introduce just a little more detail to that model. I've already mentioned that insulin is produced by the beta cells of the pancreas and glucagon by the alpha cells. These hormones then enter the bloodstream and travel throughout the body. Although they act on just about every tissue in every organ, there are three particularly important sites. First is the liver. The liver, along with skeletal muscle, is responsible for energy storage in the form of glycogen. Thus, insulin's effect in the liver is to bring excess glucose from the blood into the liver cells, where it will be converted to glycogen, fatty acids, and triglycerides. Insulin also acts in the skeletal muscle, where it promotes entry of glucose and amino acids. Amino acids are then used in the synthesis of new proteins, while the glucose is used in glycolysis to produce ATP. Some glucose may also be converted into muscle glycogen, which is energy to be used later. Finally, insulin acts in adipose tissue, where it stimulates uptake of fatty acids, which get converted into triglycerides, which can then be used as long-term energy storage. As you can see, each of these processes are reversible, and wherever insulin stimulates a process, it generally inhibits its opposite. For example, in adipose, insulin stimulates lipogenesis and inhibits lipolysis. For simplicity, I've only discussed insulin's actions here. Glucagon's actions on each pathway are essentially the opposite. While insulin and glucagon are unquestionably the two most key hormones involved in the moment-to-moment regulation of metabolism, there are many other contributors which I'll go through now. There are two other hormones to discuss that are produced in the pancreas. As mentioned earlier, amylin is produced by the beta cells and is co-secreted with insulin, and somatostatin produced by the delta cells. I'll spend a moment focusing on just how these four interact with one another. First, the secretion of insulin, amylin, and somatostatin are all stimulated by high serum glucose, while glucagon secretion is inhibited by it. While insulin suppresses glucagon secretion, somewhat counterintuitively, glucagon stimulates insulin secretion. To the best of my knowledge, this is the only interaction between glucagon and insulin that is not antagonistic. Amylin helps with insulin suppression of glucagon, while somatostatin actually inhibits both. In addition to the regulatory role within the pancreas, amylin and somatostatin have other interesting actions. For example, both slow down gastric emptying and slow down gut motility in general. This helps to blunt postprandial hyperglycemia. In addition, amylin promotes the sensation of satiety in the brain. Moving beyond the pancreas, there are two important hormones secreted by the gut. The first is called GIP, which originally stood for gastric inhibitory polypeptide. Because it's been since learned that it has minimal gastric inhibitory properties, it has since been renamed glucose-dependent insulinotropic polypeptide, a great example of a backronym in which the full name of something is created after the fact in order to conform with a pre-existing acronym. GIP is secreted by K cells, located mostly in the duodenum and upper jejunum, and its major role is to promote insulin secretion. In addition, there is the more important GLP-1, which stands for glucagon-like peptide, which also stimulates insulin release and inhibits glucagon release, as well as slowing gastric emptying, promoting satiety, and increasing insulin sensitivity in adipose and skeletal muscle. GLP-1 is secreted by the L cells of the ileum and colon. GIP and GLP-1 are known collectively as incretin hormones, or incretins for short. Incretins are hormones produced by the intestines which influence blood glucose without affecting the pancreas's exocrine function. Their existence was discovered after it was observed that study subjects secreted more insulin in response to orally delivered nutrients than in response to an equivalent amount of intravenously delivered nutrients. Thus, the stimulus for their release is dietary fats and carbohydrates. One last clinically relevant detail is that GIP and GLP-1 are both rapidly inactivated by an enzyme called DPP4, which stands for dipeptidyl peptidase. This is relevant because DPP4 makes for an interesting drug target, which will be discussed in a future video on diabetic medications. So far, all of these ancillary hormones have largely been either supporting the general effects of insulin, or in the case of somatostatin, playing more of a neutral role. However, there are three hormones that directly and indirectly antagonize insulin's actions. They are the so-called stress hormones of cortisol, epinephrine, and growth hormone. While each of these has more important primary actions, their general collective effect on metabolism is to decrease insulin secretion, increase gluconeogenesis, increase lipolysis, and to decrease insulin-mediated glucose uptake with a net consequence of hyperglycemia. You should not try to commit this diagram to memory because it can be summarized without too much loss of information. Hormones whose net effect is to decrease serum glucose are insulin, amylin, GIP, and GLP-1, and hormones whose net effect is to increase serum glucose are glucagon, cortisol, epinephrine, and growth hormone. An additional topic to discuss is the structure and synthesis of insulin. Insulin is a small protein composed of two amino acid chains linked by disulfide bridges. It's synthesized in four distinct steps. First, a single amino acid chain is formed from routine protein translation. This is known as pre-proinsulin. The signal peptide is then cleaved off in the endoplasmic reticulum, forming proinsulin. The proinsulin protein has three sections to it, designated the B chain, the C chain, and the A chain. Disulfide bridges form between the A and B chains. The C chain is then removed, which results in the final active hormone. Because the C chain serves to connect the A and B chains, it is more often referred to as the connecting peptide, or C-peptide for short. Equimolar amounts of insulin and C-peptide are stored in secretory granules and eventually released together. Therefore, C-peptide can serve as an important surrogate for insulin secretion. This is particularly true in situations in which one must distinguish between a patient who has excessive endogenous insulin present, as with an insulin-producing tumor, in which case levels of C-peptide will also be high, versus excessive exogenous insulin, as seen in factitious hypoglycemia, in which case C-peptide levels will be very low. The final topic to discuss is the temporal relationship of glucose sources. That is, where does the glucose come from that is utilized by the tissues in the hours and days following ingestion of food? In the first several hours after a meal, when the patient is in the well-fed state, the effects of insulin predominates and almost all peripheral glucose utilization draws from dietary glucose. However, after approximately 4 to 6 hours, that dietary glucose has been removed from the bloodstream, and the body's metabolism enters the fasting state. Insulin levels are diminished and glucagon increases. This results in glycogenolysis, which provides the body glucose for the next half day or so. At this point, glycogen stores are gone, and metabolism enters starvation mode. This is the point at which gluconeogenesis becomes the primary and eventually the sole source of glucose. To remind you, the process of gluconeogenesis synthesizes glucose from a combination of amino acids liberated during proteolysis, glycerol from lipolysis, and lactate from anaerobic respiration.
We'll talk about all those
While in
processes.
starvation mode, the body also directly utilizes fatty acids and ketone bodies for fuel. Ketone bodies, specifically, are discussed in more detail in the video covering diabetic ketoacidosis. So that concludes this video on the normal physiology of insulin.
Okay, so if you think, hey, why are we only talking about insulin and glucagon? It's because, I mean, we won't only talk about those, but they're really key hormones that regulate metabolism for all of our macronutrients. So they really impact how our protein, fat, and carbohydrate are used or stored. Now, we do have a third type of chemical messenger, the cytokines and chemokines, that are part of our immune system. And they basically, cytokines can alter behavior of other cells by activating gene transcription of immune cells, and chemokines induce movement of cells toward a source of a stimulus. But we won't really talk about that much in this course. We're going to really focus on the chemical messengers of hormones and some of neurotransmitters. So here's another graphic from your book that I thought was really good. And basically the take-home message here is just that there's many signals that come from your adipose, pancreas, and GI system. You can see these can be nutrients, and from your food. These can be nutrients, these can be hormones, and all of these together impact the regulation of food intake that is perceived in the brain, and that then regulates this hormone cascade, which then is going to impact our food intake level and our energy expenditure. So when we talk about chemical messengers, we have a lot of things going on. We have a lot of different signals that can result in impacts on the cells, and we can have a single hormone that can have a lot of different impacts on a lot of different kinds of cells. So we talked about transport proteins. Now we're going to talk about receptor proteins and intracellular signaling. So when we're talking about chemical messengers, we're really talking about intercellular signaling from one stimulus that is farther away to a different cell. But then when we talk about receptors, what happens? Once a chemical messenger hits a receptor, then what happens? Does the impact stay right at the cellular membrane? No, it goes within the cell. So now we have to talk about the signaling from the receptor in the cell to the internal environment of the cell. Now we know that receptors are highly specific for the chemical messengers that dock on them. They face the exterior of the cell because, of course, they need to get the message from outside of the cell. And I have a little sub-point there about oligosaccharide chains serving as recognition markers, and we'll talk more about that. But basically what that means is that on some of these proteins, there's these little carbohydrate chains that help the messengers recognize the specific cell. These receptors have an attachment site for this external stimuli or chemical messenger, and these chemical messengers can also be called ligands. And these can, as we've talked about, be hormones, they can be growth factors, they can be antibodies or lipoproteins or nutrients. So these chemical messengers can be really broad in nature. Now, when you talk about receptors on the cell and receptor types, we have three main types. We have ion channels that can move ions in and out of the cell. We have kinases. Now, get used to kinases because we are going to talk about kinases a lot, and I'll talk about exactly what they are in a moment. And then we have second messenger cells, and second messengers are the most common. And basically what a second messenger is you can just think of it as the first messenger is the hormone or the external stimulus, and then that has an impact on another either enzyme or protein inside the cell, and then that creates a downriver, I guess you'd say, or down the path, another reaction. I have a visual. I think it'll be more clear when I show the visual. But basically, you can think of second messenger steps as having multiple steps from the chemical messenger to the outcome in the cell. Okay, so second messenger. So let's focus on that because it's the most common. So the most common of the second messengers, which is the most common of the chemical messengers, is cyclic AMP or CAMP. And that should be really kind of a familiar looking molecule to you by this point. So cyclic AMP is created from ATP. So what the second messenger is, second messenger in stimulation of target cells by hormones. So basically where we get CAMP from is when we have ATP, which we know is our kind of molecule for holding our energy in our bodies. ATP encounters the enzyme adenylate cyclase, and then we get CAMP, okay? And so our first messenger, again, would be our hormone or our chemical messenger, and then CAMP becomes our second messenger. So it's really CAMP. Once it's inside the cell, it's the CAMP that's actually having the impact on the biological processes in the cell. So step one is that the ATP becomes CAMP, okay, so this is the first step. And then the step two of the second messenger is that this CAMP then activates a protein kinase. Now, as I said, kinases are going to come up over and over and over again. And whenever you hear kinase, the first thing you need to think about is phosphorylation, okay? So that's a very big word, phosphorylation, and all it means is adding a phosphate group. Now, as we'll see throughout this course, phosphate is a really common language in metabolism, okay? So, I mean, we see this with ATP, okay? We see all the time that the ATP is transferred to an ADP plus an additional phosphate, and then the phosphate's put back on and we get ATP. So we can see that just in that very simple reaction of ATP to ADP, that phosphate is having a big impact on how this molecule works. Well, phosphate has an impact on a lot of molecules, especially proteins and especially enzymes, and it can turn these proteins and enzymes on and off. So here's kind of a very generic example of a kinase that is phosphorylating. So a protein encounters a kinase. The kinase adds a phosphate onto the protein. So when you see kinase, think it's adding a phosphate onto the protein, and that phosphate comes from an ATP, which then becomes ADP. And then, so every time a kinase is adding a phosphate, a kinase is adding a phosphate. Sometimes that makes the protein active, sometimes it makes it inactive. And then, to counter our kinase, we always have a phosphatase, okay? And it just does the opposite. A kinase puts a phosphate group on, a phosphatase takes it off, okay? So that's just kind of an overview of the kinases and phosphorylation, dephosphorylation. We're going to talk about this more. So, when we have our second messenger, which again is our most common chemical messenger, what happens is that ATP becomes CAMP, and then CAMP activates a protein kinase, which then phosphorylates an enzyme to convert it to an active or inactive form. Let's look at a graphic. So on the my left here, we have kind of a generic example of a second messenger system, okay? So here we have our messenger. Think of that as the first messenger. It lands on the receptor, and which is attached to this G protein. And I think the G protein is just thought of as kind of a generic protein you can think of it as. However, this GDP is then transformed in a way that it activates this adenylate cyclase, okay? So, and then the adenylate cyclase transforms the ATP to CAMP. Okay, so the CAMP then becomes our second messenger. We had our first messenger up here, that's our hormone. What happened was that downstream it created CAMP, which is our second messenger. And as we saw in the previous slide, CAMP activates a kinase, okay? And that kinase adds a phosphate onto a protein and elicits a response in the cell. We're going to see this over and over and over again. Now let's look at a specific example. Epinephrine. We know that's a stress response hormone, right, for a neurotransmitter. Okay, so the epinephrine docks on its receptor, activates this G protein, which eventually activates this adenyl cyclase. The adenyl cyclase transforms ATP into CAMP. So the epinephrine was our first messenger. The CAMP becomes our second messenger. That activates this kinase, protein kinase A, and that's what actually has the impact of the cell. So that inhibits glycogen synthesis and promotes glycogen breakdown. Okay, so let's just think about this for a minute, because that seems like kind of overwhelming, right? So let's think about this. epinephrine is a stress hormone. Does it make sense that it's going to inhibit glycogen synthesis and promote glycogen breakdown? Yeah, it does make sense, because when we are in stress, our body is saying, okay, break down, give me energy, fight or flight, I gotta get out of here. So it's saying, I need energy. So it's gonna break down the glycogen to get you the glucose. So it makes sense. Okay, so this is, again, just reviewing what we just talked about, the phosphorylation, dephosphorylation processes. And again, I just want to drive this home because it's kind of a language of metabolism. So again, we have our protein and an ATP. It encounters a kinase. The kinase moves a phosphate from the ATP onto the protein, so it becomes the phosphorylated version of the protein plus an ADP. And then counter is the phosphatase, which takes it off. And having this phosphate on it is going to either make this protein active or inactive. So think of an enzyme. All right, so we talked about our receptor proteins. We talked a little bit about transport proteins, but let's talk more about transport proteins. So transport proteins regulate the flow of nutrients in and out of a cell. So in some cases, when we have water-soluble molecules, such as like oxygen or carbon dioxide, they can just kind of go diffuse across. But sometimes we have molecules that actually need a transporter to get them across. Now, I think we all remember our passive and our active transport. So with passive transport, it's meant for molecules that can't just diffuse across the lipid bilayer. They actually need help or a transport system to get across the lipid bilayer, but it doesn't take energy when it's a passive diffusion. However, we also have transport proteins for active transport, okay? So when we have active transport, what is happening is that a nutrient is being moved from an area of lower concentration to higher concentration. So that doesn't make sense, right? In nature, usually molecules move from areas of high concentration to low concentration so that it balances out. But in some instances, we need to move it from areas of low concentration to higher concentration. So in these instances, we need a method of doing that. In order to move against a concentration gradient, we need a pump. And in most cases, again, we'll see this throughout the semester, we're going to use a sodium-potassium pump, okay? So what the sodium-potassium pump does is it pumps sodium and potassium against their electrochemical gradient, and then that potential is used to transport other items into the cell. So in metabolism, the ones we think of the most in this way are glucose or some amino acids. So let's look at, this is just a very short video of how an sodium potassium pump works. And then we'll look at how it is used with glucose to get glucose into the cell.
The sodium-potassium pump is an active transport mechanism. 3 sodium ions bind to the protein channel, and an ATP provides the energy to change the shape of the channel that in turn drives the ions through the channel. One phosphate group from the ATP remains bound with the channel. The sodium ions are released on the other side of the membrane outside of the cell, and the new shape of the channel has a high affinity for potassium ions, and 2 of these ions now bind to the channel. This binding again causes a change in the shape of the protein channel, and this conformational change releases the phosphate group on the cytoplasm side. This release allows the channel to revert to its original shape, and as a result, the potassium ions are released inside the cell. In its original shape, the channel has a high affinity for sodium ions, and when these ions bind again, they initiate another cycle. The important characteristic of this pump is that both sodium and potassium ions are moving from areas of low concentration to areas of high concentration. That is to say, each ion is moving against its concentration gradient. This type of movement can only be achieved by the constant expenditure of ATP energy.
Okay, so who cares, right? But it is important in metabolism, very important in metabolism, because we need this type of pump in order to get certain nutrients like glucose and amino acids into our cells. So here's a good graphic of it. So here's, it's kind of backwards here, but here's our sodium-potassium pump, okay? So it's using energy to pump the sodium against its gradient. So in nature, why would the sodium want to go here? It's in higher concentration up here and lower here. So you'd think the sodium would want to stay here, right? So we need energy to pump it out. So this ATP is being used to pump sodium out and potassium in, okay? Now, once this, so this creates an even greater electrochemical gradient because it's in such, the sodium is in such high concentration on the outside of the cell. Now, we have this, we can use this energy because the sodium wants to go into the cell. It wants to go into an area of lower concentration, right? So then we can use that energy, we can use that electrochemical gradient in this symport so that it can take the sodium and the glucose from outside of the cell and move it into the cell. So it helps us move our nutrients. This pump helps us move our nutrients into the cell. Okay, so we've talked about transport proteins and receptor proteins. Now let's talk about catalytic proteins or enzymes. So the functional property of these enzymes does rely on coenzymes. And again, just like the receptor, they're very specific. Now, enzymes are reliant on how much substrate is available and how fast the enzyme can work, okay? So, I mean, we could have, if we have, let's say, all of the insulin in the world, but our receptors aren't working, well, then the velocity of how that enzyme works isn't gonna, is gonna be impacted. So it really, the concentration of these enzymes and the concentration of the substrate really impact how these biological processes go. And most of these processes are reversible, and that makes sense. We want to maintain homeostasis. So just like it showed in that video with kind of the teeter-totter, it's kind of, the reactions have got to go one way until you get too far, and then they've got to reverse. So almost all of these reactions are reversible. Although some are unidirectional, but few. So from the book, I just took this quote: many proteins and enzymes are regulated by many different mechanisms. So this is a really important take-home message, okay? So we have this one enzyme, right? Let's just think about this one enzyme, and it can be regulated by a lot of different mechanisms. So I can have a lot of different inputs from many different environmental factors, and they can all control this one enzyme, right? So this gives us a real advantage in metabolism because we can have all these different things happening and have it have the same effect, have them impact the same enzyme, and vice versa. Okay, so enzymes do regulate metabolic pathways, including covalent modification of enzymes via hormone stimulation. And again, the language of this is typically the removal or addition of a phosphate group, and especially using CAMP as a second messenger. We also have, enzymes also regulate metabolic pathways through modulating allosteric enzymes. And allosteric just means that it's relating to an alteration in the activity of the protein by the binding of a molecule at a specific site. So, other than the catalytic site. So the modulators can bind to another site and change how that protein looks, how that enzyme looks, and that can either promote or inhibit the activity. They can also, enzymes also, increases in enzyme concentration by induction is another way that enzymes regulate the metabolic pathway. And in this way, they create changes in the concentration of the enzymes, but in that way, it's typically those are unidirectional. Okay, and here we have our discussion questions. So we have talked a lot today, and if you're feeling confused, don't worry. The goal of this talk was just to introduce a lot of these concepts that we're going to see throughout our discussions on metabolism. And these are really kind of some of the most challenging concepts that we need to grasp in metabolism. So I really just wanted to give you an overview, expose you to these ideas, and you're going to see specific examples throughout the semester. And also remember, that's the metabolism aspect. Yes, we're talking the most about that, but there's also other aspects such as the digestion and transport and so on. So here's our new discussion questions. Go through them and make sure that you understand the answers. And if you have specific questions, you can you can either email me, of course, but also what might be more helpful to your classmates is if you post your question in the question discussion board of this module. So go through these questions and make sure you understand them. But again, don't get too wrapped up in the details. Just try to grasp these overall concepts. All right, I will talk to you all later. Thank you and bye.